We're going to cover every single detail you need to absolutely ace this topic. We've mapped this explainer out into 5 key steps. We'll start with mapping human genomes, move into genetic screening and diseases, unpack the genetic engineering toolkit, look at recombinant DNA and crops, and then wrap it all up with gene therapy and stem cells.
All right, Section 1, Mapping Human Genomes. This is all about understanding the complete set of genetic material.
So what exactly is a genome? For your exam, you need the precise definition and those massive numbers. A genome isn't just the active genes that code for proteins. It is the entire, complete set of genetic material. And yes, that includes all that non-coding DNA too. In humans, you're looking at a staggering 3 billion base pairs. But interestingly enough, within all that code, there are only about 20,000 to 25,000 actual genes. Think of it this way. Comparing the Human Genome Project to the 100,000 Genomes Project brilliantly illustrates how foundational science paves the way for targeted medicine. The original Human Genome Project was this massive international effort from 1990 to 2003. Because they had to analyze billions of base pairs from scratch, they relied heavily on automated sequencing machines and the brand new field of bioinformatics to store all that massive data in public databases. Following that huge success, the UK launched the 100,000 Genomes Project. But this time, they took DNA specifically from NHS patients. By comparing normal cell DNA to cancer cell DNA, for instance, scientists can now pinpoint the exact, specific mutations driving a patient's individual tumor.
Now, for the exam, you must be able to critically balance the incredible medical advantages against some pretty serious ethical dilemmas. Medically, it's a total game changer. Mapping genomes lets us identify specific disease genes, like finding the CFTR mutation in cystic fibrosis. It also birthed pharmacogenomics, which is a big word that just means doctors can tailor drug dosages to your specific genotype, maximizing how well the drug works and reducing side effects. Plus, comparing DNA helps us map out evolutionary relationships. Simply put, the more similar the DNA, the closer the relation. But as a biologist, you have to impartially evaluate the ethics here. If your entire DNA sequence is on file somewhere, there are massive privacy concerns about data leaks. Could employers or life insurers discriminate against you just because you have a genetic predisposition to a condition? And on a personal level, simply knowing you carry a risk for an incurable disease could cause severe psychological stress and anxiety.
Moving right along to Section 2, Genetic Screening and Diseases. Here's where we examine faulty alleles and inheritance patterns. You'll need to completely memorize this trio of diseases, especially contrasting the autosomal recessive nature of CF and thalassaemia against the dominant late-onset nature of Huntington's. Let's break it down. First up, cystic fibrosis. This is caused by a mutation on chromosome 7. It creates defective chloride channels, which reduces water movement by osmosis. The result? This incredibly thick, sticky mucus that blocks the airways in the lungs, causing recurring infections, and blocks the pancreatic ducts, stopping digestive enzymes from reaching the small intestine. It's autosomal recessive, meaning you need 2 copies of the faulty allele to have the disease. Next is Huntington's disease. Now this one is autosomal dominant, meaning you only need 1 faulty allele to inherit it. It's caused by expanding CAG trinucleotide repeats in the HTT gene, leading to progressive neurodegeneration. But crucially, the symptoms don't usually appear until someone is between 30 and 50 years old. This creates an absolute ethical nightmare. Do you get tested for a fatal, incurable disease you might have already passed on to your kids before you even knew you had it? Finally, thalassaemia. This is an autosomal recessive disorder that causes defective globin chain production. That leads to reduced hemoglobin, severe anemia, and chronic fatigue. It's usually treated with frequent ongoing blood transfusions. Notice how the syllabus actually requires you to impartially weigh the exact pros and cons depending entirely on when the genetic screening happens. Pre-implantation genetic testing, or PGTM, screens embryos during IVF. Now this is amazing because it prevents severe suffering by ensuring only embryos without harmful alleles are implanted. But it means the unused embryos are destroyed, which is a major moral issue for those who believe embryos are potential human life. It also inevitably raises those fears of designer babies being selected for non-medical traits. On the flip side, adult genetic testing allows for informed family planning and earlier lifestyle changes, but it brings right back those serious risks of insurance discrimination and profound psychological anxiety we touched on earlier. Okay, let's step out of the clinic and into the lab for Section 3, the Genetic Engineering Toolkit. This is how we amplify, separate, and cut DNA. Think of PCR, or polymerase chain reaction, like a molecular photocopier.
It's absolutely essential when you only have a tiny microscopic DNA sample, like maybe a single hair at a crime scene. To make it work, you need your template DNA, free nucleotides to build the new strands, and primers, which are short nucleotide sequences that bind to the target DNA and give the polymerase a starting point. And before we look at the steps, make sure you know exactly why we use Taq polymerase. It's because normal human enzymes would just literally cook and denature at the high heats required for this process. This cycle repeats 30 or more times for exponential amplification. And listen, you must cite these exact temperatures in your exam answers. First, denaturation at 95 degrees Celsius. This intense heat breaks the hydrogen bonds, physically separating the two DNA strands. Next is annealing at about 55 degrees, where the mixture cools just enough for those primers to bind to their complementary sequences. Finally, extension at 72 degrees. The heat-stable Taq polymerase gets to work adding nucleotides, synthesizing the new complementary DNA strands. Because the amount of DNA doubles with every single cycle, you get massive exponential amplification in a matter of hours. Once we've amplified the DNA, we sort it using gel electrophoresis. Think of this as an ingenious molecular sieve, where smaller pieces win the race. DNA is loaded into tiny wells in an agarose gel, all submerged in a buffer solution. Now, DNA is naturally negatively charged, right? So when you apply an electric current across the gel, the DNA is pulled toward the positive anode. Because the agarose acts like a thick, tangled sieve, the smaller DNA fragments move much faster and travel farther down the gel than the larger, clunkier ones. Afterwards, you just stain the DNA, pop it under UV light, and compare it against a known DNA marker. This whole step-by-step workflow explains exactly how crime scenes and paternity tests are analyzed. It all relies on the incredible fact that these non-coding patterns are totally unique to you, unless you happen to have an identical twin. We extract the DNA, amplify it with PCR, and then introduce a brand new tool, restriction enzymes. We use these enzymes to cut specific sections of non-coding DNA, known as VNTRs or STRs. These sequences vary wildly from person to person. We then run those cut fragments through our gel electrophoresis sieve and compare the resulting totally unique banding patterns. Now, a quick note on those restriction enzymes. Keep in mind they actually naturally evolved in bacteria as a defense mechanism to chop up invading viruses, but we exploit them for our own lab work. They cut DNA at very specific recognition sequences. Some make straight cuts, leaving what we call blunt ends. But the truly useful ones, they leave sticky ends, which are overhanging unpaired bases. These are incredibly valuable because they act like genetic Velcro. They easily base pair with complementary sequences, helping us seamlessly glue different DNA fragments together. Which brings us nicely to Section 4, Recombinant DNA and crops. Let's look at how we go about combining DNA from entirely different organisms. This right here is your classic 6-mark exam question, detailing exactly how we turn everyday bacteria into identical, low-allergy insulin factories. Recombinant DNA is literally just DNA joined together from different organisms. Here is exactly how we make human insulin. First, we isolate the human insulin gene. Second, we extract a plasmid, which is just a small ring of DNA, from a bacterium to act as a vector to carry our gene. Third, and this is the vital part, we cut both the human insulin gene and the bacterial plasmid with the exact same restriction enzyme. This ensures they have perfectly complementary sticky ends. Fourth, an enzyme called DNA ligase glues them together, forming a recombinant plasmid. Fifth, we perform transformation, which is inserting that newly engineered plasmid back into a host bacterium. And finally, we just let the bacteria do what they do best, divide rapidly, turning out vast amounts of reliable, cheap human insulin. Of course, we can use this exact same technology on plants too. But as always with biology, you must be prepared to objectively evaluate the massive agricultural advantages against the potential ecological risks. For instance, GM tomatoes have been modified to delay softening, giving them a much longer shelf life at the supermarket. GM soya has been modified to be resistant to herbicides. This is huge because it allows farmers to spray whole fields to kill weeds without harming the crop itself, massively increasing their yield. The advantages are crystal clear: more food, less crop disease, and potentially less toxic pesticide use. But you've got to balance this neutrally in your exam. Could these modified genes accidentally transfer to wild plant species? Could we inadvertently engineer resistant superweeds? And honestly, what are the unknown long-term effects on complex ecosystems and natural biodiversity? You've got to present both sides. And finally, Section 5, Gene Therapy and Stem Cells. We are looking at replacing faulty genes and repairing tissues. Gene therapy aims to replace faulty genes by introducing healthy, functional ones, usually delivering them using modified viruses or fat vesicles called liposomes. Now, if an exam question asks you about treating a disease like muscular dystrophy, you must clearly distinguish whether the therapy affects only the patient or their future children. Somatic cell therapy only affects the patient's body cells. It treats the root cause for that specific individual, but the faulty gene is still in their sperm or eggs, so it could still be passed on. Germline therapy, however, introduces the gene directly into the gametes or early embryos. That means the change is inherited by all future offspring. While this has the incredible potential to eradicate genetic diseases from a bloodline forever, it raises colossal ethical concerns about us permanently altering the human gene pool. Plus, practically speaking, gene therapy right now is incredibly expensive, can trigger dangerous immune responses, and still has uncertain long-term effects. Stem cells are basically blank canvas, undifferentiated cells that can specialize into other things. The crucial point for you to remember here is understanding their distinct cellular potential, pluripotent versus multipotent. Embryonic stem cells, which are extracted from early-stage embryos called blastocysts, are pluripotent. This means they have the almost magical ability to form almost any cell type in the entire human body. Adult stem cells, on the other hand, like the ones found in your bone marrow, are multipotent. They are much more restricted and can only differentiate into a limited menu of cell types, like various red and white blood cells. The potential uses for stem cells are just staggering, repairing damaged heart tissue, replacing whole organs, and treating severe degenerative diseases. But even with the miraculous potential to basically grow new organs, we can't ignore the massive clinical and ethical hurdles. Extracting embryonic stem cells inherently results in the destruction of the embryo, which draws intense religious and moral objections from those who view it as destroying potential human life. And biologically speaking, there's a major catch. Because stem cells are literally programmed to divide rapidly, there is a very real, very dangerous clinical risk that introducing them into a patient could result in uncontrolled cell division, inadvertently producing tumors. Everything we've just talked about, it is all pointing toward a future of truly personalized medicine, entirely dictated by your specific DNA. The days of one-size-fits-all medicine are rapidly ending. Genomics drastically improves disease prediction, allows for much earlier life-saving interventions, and ensures that the specific drugs you're prescribed are chemically targeted to work perfectly with your own unique genetic profile. You now understand the profound mechanics of how we inherit traits, how we screen for devastating diseases, and the precise molecular tools we use to literally cut and paste the code of life. But it all comes down to a choice. We now hold the pen. How will we ethically choose to write the future of human biology? Keep reviewing these core concepts, impartially weigh those ethical boundaries we discussed, trust the process, and you are going to absolutely smash this A-level module. Thanks for joining me on this explainer, and good luck with your revision.